Hello, students. I hope you're doing well and you are aware and prepared for the quiz coming right up on Unit 2. So in this video, I'm going to talk to you a bit about study techniques and also some content in Unit 2. Remember that the discussion is an excellent place to study. Look, we have 128 posts. There are 50 students in this class, so that's pretty good. Everyone's made at least 2 posts, but you need to make 3 reply posts and 1 original post. And if you've missed this deadline, that doesn't mean that you cannot do it. While you're studying this weekend, you can make reply posts as you read through the discussion. So please do that. Discussion is like collecting attendance points for coming to class, and it's a great way to interact with the material and hear from your fellow students, their own voices, and be in a community of learning together. So please come to the discussion and finish out your requirements if you have not finished them yet. So if you click here on the rubric, what I'm specifically saying is that right here you need to have 1 original post and 3 follow-up posts, okay? And we've missed those deadlines, but it's a 2-point deduction per deadline missed and a 0 or 3 points lost if you miss a post. So go ahead and do that while you're studying. Okay, now let's look at the textbook. So in Unit 2, we're working on cell division and genetics, and we're keeping pace very nicely in the labs, I feel. Keep in mind that we're catching ourselves all up on this coming up due date. So, and I'm talking to fall 2019 students specifically about the deadlines, but we'll get into more general content in just a second. So here we are in the textbook. Oh, whoops. What I wanted to point out right here is, look, you've got a lot of options for a quiz, or for a unit. And so all these numbered parts, you know, these are all the way up until 6-5, right? This is the, right here, this is the meat of it. It's not that much to read, right? And then here we get into key terms. And this is something you can study from, right? You could literally hold up a piece of paper to your screen and cover the definition and go down and see if you know the definitions of these words. And then this chapter summary is a great thing to read several times. It's not going to give you all of the detailed information, but it's going to keep your mind like wrapped around the main concept. And when I approach a textbook, like when you go to start Unit 3, I would read this first before I read the actual sections. Then here we have all these questions. So people email me and they ask, what should I do to study? Well, you absolutely should answer these questions because a lot of them are potentially in the test bank, and then also it's just great practice to frame the material in the way that it is framed during evaluation. Then there's review questions, and oh, we just looked at those, and the critical thinking questions. So what should you do? You should do those questions to study. So I want to go through some of the content. So in this unit, we're really talking about how the cell reproduces. So bear in mind what we've already learned. We've all come to terms with the idea that all living organisms are made of cells and that those cells contain the same DNA, autotrophs, same DNA and organelles. And when I say same DNA, what I mean is that the alphabet is the same. The nucleotides A, T, C, and G are all the same. And furthermore, the organelles are the same. Autotrophs have a few extra organelles to facilitate photosynthesis, but other than that, we are all remarkably similar. So as we move forward, we think to ourselves, what does it mean to be alive? Okay, and there are 5 things that define life. All living organisms can grow, they can reproduce, they can repair themselves, they can metabolize, so synthesize chemicals and make use of them. And finally, the last thing that all living organisms can do is they maintain homeostasis. So to maintain an environment within the body that is different than the environment outside of the body. So how does this happen? Let's think about what these mean. We're going to focus specifically on growth, and then we will focus on reproduction. So when we think about what it means that all organisms have the ability to grow, we're really talking about how cells divide themselves. So right after fertilization, you have 2 gametes fused into a zygote. That zygote then undergoes cell division, and after several rounds of cell division, the undifferentiated cells start to differentiate into different forms. And we'll talk about this more a bit in a later unit when we're looking at reproduction. But right now we want to think about how the cell itself divides. So in the cell cycle, it's called mitosis. Most of the time, cells are in interphase. Okay, and this is a phase when the cells are basically deciding if they're going to divide. And then once the division process starts, we go through these phases: prophase, prometaphase, metaphase, anaphase, and telophase, and finally cytokinesis, which you should be familiar with from middle school and high school biology. So what I want to do is take a moment to think about some easy tricks to remember how to work on mitosis. Okay, so when we are in prophase, when we enter the mitotic cycle, the DNA is already duplicated, and most of the time the DNA is not looking like the X's that we use to represent them in our visual models. Most of the time, it's just a bowl of spaghetti. So in the class, I draw this like this. Okay, so here's the DNA in its uncoiled state. But then in prophase, we start to see a condensation of that DNA. So in some of these, you can see how, their stain it's more dense in certain areas inside the nucleolus. And so that's the chromosomes actually condensing. But then here's a cellular image of, sorry about that, anaphase. And see here, you can see the condensed chromosomes very easily. All right, so enough about the model. Let's go back to our page in the textbook. Okay, so back to this image that really contains almost all the information for this entire chapter, so we are going to dwell here. Essentially, we have the bowl of spaghetti, and then late in interphase, the DNA duplicates and the chromosomes start to condense. And then we are always going to be thinking about them as these X's. And we're going to want to think about when the X's divide and when they duplicate, etc. So in prophase, the nuclear envelope starts to disintegrate, and the cytoskeleton really changes. One thing that I never appreciated really when I first learned about mitosis is how important those microtubules are to what the movement that's occurring in mitosis. So essentially, the chromosomes condense in prometaphase, and these kinetochores appear at the centromeres. That's just right in the center of the duplicated chromosome, so you can see this little dot here. And then we have the mitotic spindle microtubules attaching to the kinetic cord. So what I want you to think about is that these lines that are in the cell, this is like a chemical reaction that's occurring through a domino effect. So I liken it to like a comic flip book, or, releasing a string of dominoes and watching them fall. So what's actually going to be pulling these chromosomes apart as we move through the phases is a mediated chemical reaction. It's not just happening without any chemical reaction behind it. So here in metaphase, I like to call metaphase meet a phase, right? Because the chromosomes are meeting at the equatorial plate, right in the middle of the cell. And right now, see, we're talking about how these are sister chromatids, right? These are copies of the chromosomes. And then as we go into anaphase, sorry, I'm having a hard time deciding how big this should be. Move this up. Nope, can't do that. Then as we move to anaphase, the sister chromatids, which are now called chromosomes, are pulled towards opposite poles through that like comic flip book chemical reaction. And then as we move to telophase, we see like this reformation. So things start to rebuild. And so the chromosomes and the duplicated cell contents, including the organelles, are all at the poles of the cell. The nuclear envelope starts to surround each set of chromosomes, and that mitotic spindle starts to break down, and we go back into a normal cytoskeletal situation. Okay, and then finally cytokinesis, that means like cell birth, right? In an animal cell, which we're viewing right now, we see this is called the cleavage furrow. And in a plant cell, you can see that it's more angular, right? So the cell wall extends between, and then you have two cells. And we'll look at the microscopic images in just 1 second. So this is mitosis. And essentially what I want you to understand is that everything has to be duplicated, it has to migrate to different poles, and new cells are created. And this process is how your body is growing at all times. Certain kinds of cells, like your epithelial cells, your skin cells, they are regenerating on the outside all the time. Other cells don't regenerate, like most of your neurons in your brains are not regenerative, but the neurons in the hippocampus are. That's why we're able to learn continuously throughout life. So that's mitosis. Let me get back to my microscopic image for 1 more second. Okay, so this picture, by the way, I took this picture with my phone, holding it up to the eyepiece at the microscope in the lab. So it's a pretty good picture, I think, but you can play around with that if you're using a microscope. So take a look here. In this image, we have all the phases represented. So we talked about like the bowl of spaghetti, wherever, there's like an equally dense but not very dark center nucleus. That's just interphase. And then as the chromosomes start to condense, they become darker. And then this is a little foggy right here, but here we see metaphase, where the condensed chromosomes, the sister chromatids, are lined up on the metaphase plate. And then over here in anaphase, we're seeing that these sister chromatids separated are now called chromosomes, moving towards the opposite poles of a cell. And then everything starts to reform. And so right here you see kind of this anaphase is probably right here. So you can't see the cell wall is not continuous right there. But then on this one you can see cytokinesis. They're both there. And so here we have it. Please appreciate that if we weren't looking at these diagrams, this image, which is not a model, it's just the best we can do at looking at this, doesn't impart as much information. Most importantly, it just doesn't show us anything about the mitotic spindle and how the centromeres work and how it's chemical mediation that pulls these materials to opposite sides of the pole. All right, in the next video, I'll talk about meiosis.